Enzymes. Introduction. Recall from your previous classes that enzymes are biological catalysts which increase the rate of chemical reaction in living cells without being used in the process. Nearly every chemical process that takes place in living thing is facilitated by an enzyme. The sum of all the chemical reactions that a cell or larger living carries out as its metabolism. Many activities in living things are controlled by metabolic pathways in which a series of interrelated steps are involved, each one of them facilitated by an enzyme. Enzyme structure. Enzymes are proteins and their function is determined by their complex structure. This reaction takes place in a small part of enzyme called the active site, while the rest of protein acts as framework. The amino acid around the active site attach to the substrate molecule and hold it in position while the reaction takes place. This makes the enzyme specific for one reaction only, as other molecules would not fit into the active site. Mode of enzyme action. Following are two hypotheses explain mode of enzyme action. Log and key hypothesis. Fischer in 1890 suggested that each enzyme had a particular step in which the substrate fit exactly. This was known as the lock and key hypothesis. According to this hypothesis, the substrate is imagined like a lock while the enzyme is imagined like a key, as one specific key can open only a specific lock. Similarly, a specific enzyme can break up only one specific substrate. The active site is regarded as rigid structure that does not modify or change during the reaction process. However, later studies did not support this hypothesis in all types of reactions, and therefore the hypothesis was modified into induced fit hypothesis. Induced fit hypothesis. The attraction of the substrate and enzymes form an enzyme-substrate complex. It was originally referred to as the lock and key enzyme theory. The current theory suggests that the enzyme molecules are in an active form. To become active, they must undergo a slight change in structure to more specifically accommodate the substrate. It is said to be induced with substrate. Think of where your hand changes shape slightly when you shake a person's hand. There are three ways of thinking about enzyme catalysis. They are described the same process, though in different ways, and you should know about each of them. Reaction mechanism. In any chemical reaction, a substrate is converted into a product. There may be more than one substrate and more than one product, but that doesn't matter here. In an enzyme catalyzed reaction, the substrate first binds to the active site of the enzyme to form an enzyme substrate complex. Then the substrate is converted into product while attached to enzymes, and finally product is released. This mechanism can be shown as in figure 3.2. The enzyme is then free to start again. Energy changes. The way enzymes work can also be shown by considering the energy changes that take place during a chemical reaction. We shall consider a reaction where the product has lower energy than the substrate. So the substrate naturally turns into product. In other words, the equilibrium lies in the direction of product before it can change to product. We shall consider a reaction where the product has lower energy than the substrate, so the substrate naturally turns into product. In other words, the equilibrium lies in direction of product before it can change into product. Substrate must become energy barrier called activation energy. The larger activation energy, the slower reaction will be because only a few substrate molecules will by chance have sufficient energy to overcome the activation energy barrier. Enzyme dramatically reduce the activation energy of reaction so that most molecule can easily get over activation. Cofactors. Cofactors are atom groups of atoms and molecules that join with enzymes, altering their shape and making them functional. One can think of these cofactors as an on-off switch for activating an enzyme. If the cofactor is non-protein like a metallic ion, zinc, copper, iron, it is referred to as phosphatic group. Phosphatic group. Some cofactors are small organic molecules called coenzyme, like enzyme. They are not permanently altered in the reaction. Many of these coenzymes are divided from vitamins and minerals that are essential for life. The absence of these cofactors can lead to vitamin and mineral deficiency diseases. For example, lack of vitamin B produces beriberi. Example of coenzymes are NAD, FAD, and ADP. Enzyme nomenclature. Many enzymes, but not all, end in the suffix "se", exception pepsine, trypsin. They are named for the substrate that act on the action they perform. The following are the six major enzyme categories. Oxidoreductases. These enzymes catalyze various types of oxidation reduction reaction. Subclasses of this group contain oxidases, oxygenases, and for oxidases, transferases. These enzymes catalyze reactions that involve the transfer of group from the molecule of another. Examples of such groups include amino, carboxyl, methyl, and carbonyl, transcarboxylase, and transmethylases are examples of transferases. Hydrolysis. This enzyme catalyzes the reaction in which the cleavage of bond is accomplished by addition of water. The hydrolysis include esterases, phosphatases, and peptidases. Lysases. Catalyze reaction in which group H2O, CO2, and NH3 are removed to form a double bond are added to double bond decarboxylases, deaminases, and synthesis are examples of lysis. Isomerases. This is heterogeneous group enzyme which catalyze several types of intermolecular rearrangements. Epimerases and mutases are examples. Ligases. Ligases catalyze bond formation between two substrate molecules. The energy for these reactions is always supplied by ATP hydrolysis, factor that affect rate of enzyme reactions. Rate of enzyme reaction depend on following factors. Temperature. Enzyme work best at an optimum temperature. Enzyme present in mammals works best at about 40 degrees centigrade. Animals present in different environments are adapted to range of temperature. For example, enzymes of Arctic snow flea work at minus 10 degrees centigrade, whereas in thermophilic bacteria, enzymes work at temperature of 90 degrees centigrade. Up to the optimum temperature, the rate increases geometrically with temperature. The rate increases because the enzymes and substrate molecules both have more kinetic energy, so collide more often, and also because more molecules have sufficient energy to overcome. Activation energy, the increase in rate with temperature, can be quantified as a which is the relative for 10 degrees centigrade rise in temperature. The rate is not zero at 0 degree centigrade, so enzymes still work and the refrigerator and food still goes up, but they work slowly. Enzyme can only work in ice, though the rate is extremely slow due to very slow diffusion of enzyme and substrate molecule through the ice lattice. pH. Enzymes have an optimum pH at which they work fastest. For most enzymes, this is about pH 7 to 8. Physiological pH of most cells, few enzymes can work at extreme pH, such as protease enzymes in animal stomachs, which have an optimum pH 1. The pH affects the charge of the amino acid at active site. More specifically, changes in pH ionizes amino acid forming an enzyme. So the properties of the active site change and the substrate can no longer bind. Enzyme concentration. As the enzyme concentration increases, the rate of reaction increases linearly because there are more enzyme molecule variable to catalyze the reaction. At very high enzyme concentration, the substrate concentration may become rate limiting, so the rate stops increasing. Normally, enzymes are present in cells in rather low concentration substrate concentration. The rate of an enzyme catalyzed reaction shows a curved dependence on substrate concentration. As the substrate concentration increases, the rate increases because more substrate molecules can collide with enzyme molecules, so more reaction will take place. At higher concentration, the enzyme molecule becomes saturated with substrate, so there are few free enzyme molecules. So adding more substrate doesn't make much difference. Inhibitors. Inhibitors inhibit the activity of enzymes, reducing the rate of their reaction. They are found naturally, but are also used artificially as drugs, pesticides, and research tools. There are two kinds of inhibitors. Competitive inhibitor molecules has a similar structure to the normal substrate molecule, and it can fit into the active site of the enzyme and therefore compete with the substrate for the active site. So the reaction is slower. For example, sulfonamide to an antibiotic drugs which act as competitive inhibitors. A non-competitive inhibitor molecule is quite different in structure from the substrate molecule and does not fit into the active site. It binds to another part of the enzyme molecule, changing the shape of the whole enzyme, including the active site, so that it can no longer bind substrate molecules. Inhibitors that bind weakly and can be removed out easily are sometimes called reverse inhibitors, while those that bind tightly and cannot be removed out are called irreversible inhibitors. Poison like cyanide, heavy metal ions, and some insecticides are all non-competitive inhibitors. The activity of some enzyme is controlled by certain molecules binding to a specific regulatory or allosteric site on the enzyme. Distinct from the active site, different molecules can inhibit or activate the enzyme, allowing sophisticated control of the rate. Only a few enzymes can do this, and they are often at the start of a long biochemical pathway. They are generally activated by substrate of the pathway and inhibited by product of the pathway, thus only turning the pathway on when it is needed. Feedback inhibition. Another kind of inhibition is called feedback inhibition. And feedback inhibition, here is a second binding site on the enzyme where the inhibitor binds so that the inhibitor is not necessarily similar in structure to the substrate. The absence or presence of the inhibitor at this second binding site activates or deactivates the enzyme by changing the conformation of the enzyme so that the active site is made available for unavailable to substrate. The inhibitor is usually the product of reaction formed during the metabolic pathway. Enzymes are organic chemical substances produced by living organisms to speed up a particular reaction, but during this process, these remain unchanged. Enzymes are very specific in their action, acting on specific substrate. When an enzyme acts on a specific substrate, enzyme substrate complex is formed. When enzyme shapes are disrupted, it loses its characteristics. Biological activity. The non-protein part of prosthetic group of an enzyme is called cofactors. According to lock and key model of enzyme, the active site of an enzyme is a rigid structure. Modification to the lock and key model suggests that since enzymes are rather flexible structures, the active site is continually reshaped by interaction with the substrate as the substrate interacts with the enzyme. An inhibitor is a chemical substance which can block the active site of an enzyme temporarily or permanently by stopping the activity of enzyme. Factors that affect the rate of enzyme action are enzyme concentration, substrate concentration, temperature, pH of medium, cellular control mechanism in which an enzyme that catalyzes production of a particular substance in the cell is inhibited when that substance has accumulated to a certain level, thereby balancing the amount provided with amount needed. After that, we will discuss chapter 4.